Okay, now we're recording. Hi, it's Friday. So we're going to analyze the gels. And this one is kind of crappy. So that's why I'm going to use it as an example of sometimes shit happens. Um, and what you can see here, or what you can't see here very clearly, is the ladder. So this lane right here would be where the molecular weight marker is. And so it's super light. And there's some different options of why that might have happened. It could have been underloaded just from the beginning. But since we don't see any PCR product bands, I'm assuming things were loaded in some of these other wells also, and we don't see any bands showing up, and that this one is super light, is that it's possible that when these were being loaded, they actually kind of jumped up out of the wells. So when you load a gel, you put that, if you remember from the Jove video, you put the gel in the little contraption, and then you pour your 1x TBE buffer, whatever the same buffer that's in the gel, on top. So it's just covering the gel. So you want the current to run through the gel, not over it. So if you have, a centimeter of buffer above your gel, most of your current's going to run over the gel and not through it. And so then it'll just take a lot longer to run your gel. But if you put not enough and it's just sort of hovering right above the wells, it can make that little meniscus. And with capillary action, a property of water, it kind of sucks it right out of your wells. So you might load it and it seems fine. And then when you go to put the lid on and it jiggles a little bit, it'll suck all your product right up out of those wells. And so that's possible that that's what happened here. So you still see some was loaded in, some of it didn't jump out, but most of it did. So you can barely see the ladder and we can't see any bands that may have come up. It could also be this PCR didn't work and that somebody over diluted the ladder when you get it. Like they didn't do they were supposed to make it a 6X or a 5X and it was really at a 1X and then people diluted it thinking it was 5X to 1X and, blah, blah, blah. So there's all kinds of different options that might have happened. But in any event, this is a picture of the 1 kb marker from Thermo Fisher that we generally use in the lab. And so under the best situation, you should be able to see 3 different, brighter bands. And you can see that this top brighter band corresponds to 5,000 base pairs. This guy's about 1,500 and this one's about 500. And then you can see the other bands, they're marked as what their sizes are. And so then that's how you figure out what size bands you have by where they might be. So this guy right here looks like a brighter guy, which is probably that one. And so there's supposed to be 4 bands below it. The smallest is 75. I only see 3, but this is also very light. And that 75 base pairs, remember when we talked about agarose gels and what can resolve and what can't? That's really pretty small, especially if this is probably a 1% gel. Maybe it's even a 0.8%. I don't know. So we might not see that bottom guy anyways. So we would assume this is about 500. And then if you use the ladder and you count up 2 more, right, there's another bright one right there. That might be the 1,500. And then you count, this gets very smeary, but there's something a little bit brighter, maybe right there, about 5,000. So when you scan through the other gels in this document, you'll see ones that are a lot clearer. So I'm using this one as an example, even though it stinks, just so that we save the other 6 that are a little bit better than what we see. And then just for practical reasons, so the pink stuff that you see is the excess ethidium bromide. So when we use ethidium bromide, we talked about we don't put it in the gel, right? Remember I made that big fit and I talked about that all during those Jove videos also, that we soak the gel in afterwards. And so ethidium will also just non-specifically suck up into the gel. And a way to actually reduce some of that pink would be to take this gel, once you soaked it in ethidium, and then soak it in plain 1x TBE. and let it sit for 10, 15 minutes or so. And that will remove some of this pink background, which will make the Ethidium that's actually intercalated into the DNA stand out a little brighter, because this will be more of a blue background versus, this pink smeary stuff that's in this guy. So it'll pull it out of the gel that's non-specifically stuck, but it won't pull it out of the DNA that's actually intercalated. Now, if you forget your gel in a destain overnight, it'll destain everything. But 10, 20 minutes in the destain, which is just plain buffer or water, if you don't care about ever running it again, will just pull out some of that background and make this foreground look a little nicer. Now, if you can see what you need to see, you don't have to do that. Like, who cares? And if this one you were pretty sure there's no bands in here anywhere, you'd be like, well, why waste my time 15, 20 more minutes and then take another picture and say, there's a really nice ladder, but there's still no bands there. So you can see in some of these other guys over here, the levels of pink. Now this one is like way overstained. Like this, either somebody put way too much Ethidium in the bath or this sat in there for a long time, but who cares, right? Except that it's contaminated and you want to be super careful because you can see the bands super clearly. So removing the pink background might be for aesthetic reasons, but for science reasons, there's no reason to spend your time doing that. Because then you also then have that destain bath, that 1XTB is now technically contaminated with Ethidium and either needs to be disposed of as a liquid waste for Ethidium, or the Ethidium removed using the little tea bag stuff, or saved for diluting a bath that's like super Ethidium. So as little Ethidium waste as you can have, the better. And if it's a gel red or if it's at cyber green, it's the same thing. You still have to dispose of that stuff as hazardous waste because we really don't know how hazardous whatever. It still could be bad even though they claim it's not as bad as Ethidium. We still wouldn't trust it. So I'm gonna just click a couple without giving away the farm. So this guy, right, has multiple bands in each lane. And so you'd want to say about how big you think these bands are. You don't have to label the entire ladder, but you might want to just put an arrow onto whichever one you think is the bright one. And if you're wrong, who cares, right? For this, I just want you to get it. Like, so just designate the 3 bright ones. And if they're not correct, who cares? It's just so that you can estimate the sizes of the other bands to whatever you claim this ladder is, because this isn't real, like we didn't do it. And so you'll tell us what size you think these 3 are and what size you think those guys are. You see this little guy up there, he's even bigger than off of what we think we can see here. And then go back and see what your PCR product's supposed to be. Like mine's supposed to be 512. So I would expect to see something around the 500. So if we were looking at my cheesy gel and I designate this one as my bright guy at 500, I would expect my band to be right here. And I don't have any bands. So I'm like, I failed. But if this were my gel and I'm expecting 500, here's a bright guy down here, and my band's way up here, I'd be like, yeah, that's so not 500 base pairs. So what would I do? Well, if I got bands, but not the one I want, I'd probably have to lower my temperature because I need it to sit on my spot, right? And the annealing temperature is the one that makes it more stringent or harder to sit down. So we need to make primers, need to make them sit down easier. Even though we got this crap up here, we don't care if this stays as long as we get a band around 500, because we can always do the gel extraction, just like we learned in our Jove videos. So right? So here's again, if that's at 500, nope, not right for me, right? And so but if your band is supposed to be 1,000, then you would want something around this band, right here at 1,000. And so then you want to look and see if any of the gels have that. This guy's big, nothing else. There might be somebody way down here, figure out what size that is. So if you want to in your group, you can just randomly pick one, say, I'll take number 3 without even looking at it. And because it doesn't matter if it has your band or not, right? It's just, can you analyze a gel so that when we do load a gel with our PCR, you'll be able to look at the picture and know exactly whether your PCR worked or not. Do we raise temperature? Do we go down in temperature? Do we do a gel extraction? Do we try again? Right? If we have lots of bands and let's say we really want this one, this is the size that we want, but we're seeing another band, then we'd want to raise the temperature. We want to make it harder for our primers to sit on things that they shouldn't sit on, right? Our sequence was 100% complementary. That's how we built our primers. But when we looked at the primer blast check and we got all our mismatches, right? Remember, those were usually off by 3 or 4 base pairs. So they shouldn't sit down on the genomic DNA as well as our guys that were 100% complementary. But sometimes they do. So if that happens, then we need to make it harder for primers to sit down. So we would raise the temperature. And what we would hope is that the bands that aren't supposed to be there will disappear and we'd be only left with the one that we want. So that would be some of the troubleshooting that you'd need to know how to do. So whether you're pipetting now or not, if you don't know how to do that kind of stuff, you're not going to get anywhere in the lab once you take that picture. So all of our upfront knowledge is really going to help us like make the experiments happen much more quickly than in a standard semester where you're pipetting and learning at the same time. So another example of, like, let's say this is your band of the size that you want. In this case, we didn't have this in our videos because of course I couldn't find one, but if you only get one nice clean band, you don't have to do a gel extraction. And that's because if you think about, we made our PCR reaction be 50 microliters and we only load on a gel about 20. That's all that can fit in these little wells. We had a couple microliters of the dye and the glycerol to make it sink. And it's about 20 microliters that you can fit in here. That means your tube has 30 microliters left in it. And so you can just run a PCR cleanup on that 30 microliters, right? It's got that same band in there. You just can't see it. It's floating around in clear liquid. And it goes through a filter. It does like that same ion exchange type stuff with pH or salts. And it essentially cleans out all the primers and a lot of the proteins in there, the enzymes, all that other stuff that might interfere with the next reaction, which is that ligation into a vector that we also learned about. So the PCR cleanup, you don't lose efficiency. You get back 90% of the DNA that you put in there. And so you're just ready to roll. Whereas if you were in a situation where where's that other one that I just did? Like this guy, where you have multiple bands, and let's say you raise the temperature and you did, a bunch of stuff to a point where you're like, okay, these bands are not going away. And if this is the one you want, you'd have to gel extract it away from this band, because this tube, the 30 microliters left over from this tube, has more of what you don't want than what you do want. And so if you just PCR cleaned it up and did your ligation and cloning into that vector, more than half, probably 3/4 of what you get are going to be the wrong thing. Now, you could just screen lots of colonies to find the right thing. But sometimes when we play that game, if you look just at the intensity, maybe this would be 3/4, this would be 1/4. But if you actually do the experiment, it ends up being 99% this and 1% this. And if you have 1,000 colonies, you might be picking 50 before you find the one that has what you want. So in that case, we'd rather take the risk of doing the gel extraction so that we can cut out just the band. We want just the DNA and do that gel extraction cleanup. And then we're just cloning this little guy, right? Versus all the stuff that's in that tube. So those are just some options that you have, you know, when you run PCR. PCR cleanup's always a better option if you can. Gel extraction gives us some issues sometimes. The agarose, if any agarose gets through the filter, that can cause blockage of that ligation reaction, sometimes the pH, and then sometimes you just get no DNA back, which totally blows. You can't clone something if you don't get anything back. So it's always a little bit, it's safer to do PCR cleanup if you can. So if you know, on your first run, you get multiple bands, I'd always futz around with the annealing temperature to try to get down to one band. But then once you've done it a few times, you could spend all semester just doing PCR to try to get one band. At that point, you're like, all right, I'm just going to risk it on the gel extraction and just try to be really careful. And then you still have that extra 30, like if your first go at a gel extraction sucks, you run another gel with as low percentage of agarose as you can, and then, you know, try to gel extract that one. So there, and if that's a disaster, you know what the PCR reaction and the products and what you need to do to get that more of that band. And so then you just do another round of PCR. You could make, use a comb with bigger wells. So you could load more like 30 or 40 microliters in a lane so that your band of interest is much brighter to cut out. So there's lots of options you can do to push your efficiency towards getting some DNA back and making your reactions work. So there's lots of fun little tricks we can do. But for today, my little friends, so you're going to want to designate at least the bright guys on your ladder, on your Google Slides after you made your copy, so you have your own very own copy. And I made it in Slides because if you've never used Google Slides, you can make, you know, arrow shapes to like point stuff at. Oh, I failed. To point at stuff. I think, can I do that? Yeah, like, ooh, I can make an arrow. You can add text, you could do all kinds of stuff so that you can, you know, show me that you know how to analyze this gel. If you want to move this guy over so that it's easier to see. Oops, I want to move to front. Do they reorder here? No, I don't know. I don't know if you can.
You have to go to arrange.
Oh, it's on, yeah. Where?
On the top where it says file, edit, view.
Oh, right there.
Yeah. And then order.
Yep. And then, yeah.
I'm
like, it's somewhere. Oops, I had the wrong one. I had the, yeah, okay. Yeah, it's similar enough to PowerPoint, but not exactly enough to, yeah, so then I could just move this guy, you know, and I can sit him right there so that it's easier. And then, you know, you can try to make this be exactly the same size. Again, you don't have to, you know what I mean? But like, there's lots of, especially like if your gel's kind of crappy and it's hard to see. All right, you can make it, you can make it line up exactly, right, to help you out. So whatever you guys need to do to make that guy, and then what you'll need to do is you can either just, you know, download this whole file and upload it to the assignment, which I have to set up during the breakout, or you could take a screenshot and just upload the screenshot, whatever, just evidence that, you know, you tried. And then I'll try to go over them also so that, you know, and if you're wrong, you're wrong. It's totally fine. Like, this is your first attempt at this. Like, we just want everybody to give it a shot versus me just telling you, so that then if you did it right, you're like, woohoo. And if you know, didn't really get it, like, it'll make more sense because you tried it on your own. So questions, I guess, before I keep yapping. We're good, my little friends. Okay, I'm gonna, oh wait, first I'm gonna say, oh, that's not what I wanted. why? Okay, maybe I have to do this first. Okay. I would like to, why am I having so many issues? But it's like I've never used Zoom this semester. Why is this? Go away. Okay. I just want to make it so you guys could share your screen with each other. Why is this not letting me pick? Oh, advanced. Okay. I don't know why. Okay. The hell? I'm not. Where? Okay, hang on. Why? Usually, okay, there we go. Gives me my little op. Okay, all participants can share. So you should be able, okay, I just selected that. So you should be able to share your screen in your breakout room with your pals so that if you want to help each other, you could share a screen and you can, you know, help. If you want to try doing it on your own before you share it and have somebody else in your group help you, unless you just can't even start. You're like, I don't even know what the hell's going on here. Then get some help to get started. So yeah, while you guys are in your rooms, it's 3s and 2s, just because that's how many of us. I will set up the assignment upload. I think I have one there, but I think I may have only made it for the Wednesday people. I don't know. I'll look at it and I'll find those other forms for next semester too. And then I'll just send you guys a couple of little messages. You know, we can take as much time or as little time. So if you guys get yours, if you get yours finished, just come back. And if you have questions, just come back and yell for me if I'm not sitting right here. Okay, I'm about to open. Are you ready? Woohoo, open! Uh-oh. Okay, that's fine. All right, go. Hey, Jared.
Hi, Professor. I just had a quick question again.
Sure.
So on Canvas, it says there's the figures and like figure legends and like lab, like meeting presentations and all that.
Yeah.
I know we talked about the presentations, but I'm not sure how to do the figures because it's not on the.
Sorry, I'm going back and looking at my.
It's not on the sample or like example write-up. So I was just wondering, are those only for people who are making posters about their project?
I can't remember what the write-up. There was no figures in the write-up.
All right, let me double check just in case.
I'm like, that's not a good example if there weren't.
There, unless it just means like the actual figure legends, which
Let me see it. I'll open it too because I don't. I mean, you know, I have it somewhere, but good Lord, I'll never find it. I'm just gonna open it in Canvas and see. Okay, so it shows their vector maps.
Mm-hmm.
Right? Yeah, so they just have their figures within the paper. So, you know, I just have.
So if I already did that part.
Yeah. Just upload like the whole thing into, you know, I just broke it up just in case people need the
like the extra time.
Yeah, if they really, really need to sorry, I'm assigning somebody that came in late to a room. If they really need that motivation to get shit done, right? I have them broken up, but you know, I'm not going to go, oh my God, they put it in their final version, but it's not in that section, you know.
Okay,
that's really just to help people like set their time.
I was just a little bit, I guess, confused because like I have the figures and then I have like the figure legends like akin to what it shows, like where it's the small little like descriptions of the figures. Like I've already done all that because I've been trying to go date and date with the.
Yeah, then just leave them in the full thing and you don't have to upload anything to that little section.
Okay. Yeah. Then I'll just kind of work ahead then because I've been trying to follow the the due dates kind of. So I'll just work ahead with like what's due in 2 weeks. Or yeah.
Yeah, I mean, you can get it all done ahead and just be done if you want to. You don't have to wait.
Thank you again, Professor.
Yeah, or you could, you know, if you need more time for other stuff, other classes right now, you know, you can skip the deadlines and just get it done at the end.
Okay, thank you, and thank you so much.
Okay, no problem.
Bye, Professor.
Bye, good to see you.